Hi everyone, I'm Corey Watson, for those of you who don't know me. I'm an associate professor at the University of Louisville, and a long-time AIRRC member. So I have the pleasure today to kind of lead us through the Germline Challenge Session, which the meetings subcommittee planned to try to cover some areas that we think we've learned things about germlines over the last 9 years, and where we kind of see this field going. I'm going to try to give a brief overview of this sort of topic at large before I introduce our keynote, William Chief. Cool. So for those of you who don't know, at least in the eyes of AIRRC, the germline sort of efforts and initiatives by our community started really at the first meeting. And the germline group, although it wasn't formed in 2015, the working group arose out of the 2016 meeting. And a lot of that focus was, at the time, centered around this idea that we were recognizing that databases were incomplete for germline genes and alleles. And yet these were really important for all the things that we were trying to do as a community. So they have continued to prove to be these sort of centerpieces of most analysis pipelines for BCR and TCRC data sets. And so the germline working group, which you'll hear more about as the session goes on, was really focused on, laser focused on this notion that we wanted to improve existing databases at the time and try to make them more complete, more accurate, and of course enriched with additional metadata. And of course, a lot of this was driven by what I like to refer to as allele envy. We know that the HLA locus has forever basically been thought to be the most diverse region of the human genome. And of course, these other really important immune loci, which we call TCRs and BCRs, or antibodies, also really important to the immune system. And we have known for a while that they are also very diverse, yet understudied. And so just to sort of pinpoint this, I'll use the TR alpha and beta B regions as an example. So for HLA, of course, in this plot, which is a couple years old now, there's somewhere over 26 class 1 alleles, whereas for alpha and beta, there are less than 300 documented today in IMGP. So I don't believe that that's an accurate representation of what's out among our population, our species, and I suspect most other people who work in this field don't either. And so that was a sort of testament to the work that we had to do. And so the germline sort of effort in this community have done a lot in the last 9 years. Sometimes it feels like we haven't gone very far, but if we self-reflect, we can see the fruits of our labor for sure. A lot of what we've done is focused in these 3 boxes here. So we've spent a lot of time trying to develop and implement new standards and policies around germline databases. And this has basically led to multiple publications and ended up by many groups in our community, the development of tools for processing germline data, inferring novel germlines from AIRRC data, and working really hard to put those policies in context and try to get these data into germline databases, is where a lot of our effort is spent today. And the goal that we all have looking forward is this purple box here, and that we see the field changing drastically. We see that the need for germline databases is not decreasing, but increasing. And so a lot of what we're trying to do today, and you'll hear others in this session talk about these things, is build up databases to set a foundation that allows us to be responsive to all the things that are happening in our sort of data landscape with all the new technologies that are coming through, so that we can continue to build even more robust data sets as we go. And of course, along the way, we started to learn a lot about human population diversity, and this continues. A lot of the work that we've seen in the last few years has told us that we are only now scratching the surface, which is both daunting and exciting. But a lot of the new techs coming down the pipeline are now going to let us start capturing more of these alleles and genes at scale. Also, we are beginning to look into a lot more additional species. Again, things like long-read sequencing are allow us to access very complex genomes, and you'll hear more about that today. And this is allowing us to not only build up databases for these species and sort of this under this sort of practical objective, but also begin to learn more about how these loci evolve through time. And I'll just say as a testament for my own sort of surprises in this field, that these genes are, I think, probably one of the most fascinating immune system genes in the genome. And they certainly have many, many more surprises for us as we go into these other species. I mean, who doesn't want to know what a giraffe antibody looks like? Got a long neck, that must mean something. We're also learning a lot about animal models, and we have learned a lot about animal models. And I think, again, many surprises here. And we still have a long way to go to represent these in databases, but trying to understand what it means to have a mouse strain, which is supposedly two strains in the same species, and how we represent these both in databases and biologically is really important. This extends to agricultural animals as well. So thinking about breeds, a cow is not necessarily a cow, and that's something that you know, we are learning how to deal with and think about more broadly. In addition, I'll say, and we're going to hear about this today as well. Everything I'm putting in here, we're going to hear about, I guess I should say, so I should quit saying that. A lot of what our focus in the last 9 years has been is really the variable regions, so looking at VD and J genes and describing their diversity. And in the process, we've left out constant region genes. For better or worse, it is what it is. But I think those of us who are starting to look in these regions of the genome now are realizing that these have their own stories to tell. They are, at least in human, from data that we've seen, on par with the VD and J regions in terms of diversity. And I think that characterizing this genetic variation is going to hopefully open up a lot of novel discoveries and antibody function. TCRs as well. You can tell I'm an antibody biased person, but TCR constant regions are also important. And then, of course, we began to learn, and we have a whole lot more to learn about actual direct germline impacts on all the repertoires that we study, including in the context of disease. And we now have a good understanding, at least, that we know germlines can influence the composition of the repertoire in terms of influencing gene usage. This is supposed to likely influence evolutionary trajectories of antibodies through a response. And of course, there are now these very direct examples where coding changes can actually modify the binding affinity of particular antibodies. And of course, we'll probably hear more about that today as we go on. And so that brings us to some of the bigger questions in the field, I think, or at least where we're going now, is beyond trying to understand how we link genetic data and AIRR-C data together. It's also how we do this to understand more about the immune response in clinical phenotypes. And I think that's going to be something that we see a lot of effort in the next few years, and I think we're gonna see that germline diversity is part of the puzzle and something that we all need to be thinking about as we analyze our AIRR-C data.